Great, we're on. So when we speak about the forensically important Caliphorids in Britain, it's mainly three subfamilies, which are the Califorinae, commonly known as the Bluebottles, Lucyilia, Greenbottles, and the Chrysominae, that are often called black blowflies. Out of Lucyilia, one species can be excluded on the basis that it is only found feeding on living amphibians such as the toads and frogs and salamanders, and this is Lucyilia buffonivora. So yes, that species has no forensic importance whatsoever, but it's still quite interesting to see. Most Caliphorids as adults feed on slightly less gruesome things. They mainly need sugar, which they get from things like flowers and fruits, honeydew, sap, and others. And especially males are pretty much happy just with this kind of diet. However, the females, they do need protein in order for their ovaries to develop and so they can achieve the sexual maturity. And that is usually gained through feeding on meat and rotting fish, other feces, and other foods that contain a lot of protein. Just as a reminder for those who did previous course with me on identification, the species here is Caliphora vomitoria, which we can easily tell by the ginger beard indicated by the arrows. Okay, so females, as already mentioned, need the protein meal, and that can be gained through feeding on blood, corpses, carcasses, and other sources of that sort, which is for forensic entomologists not always a welcome situation, because females are regarded as difficult, and difficult in terms of identification, which hopefully, since the publication of the new key, is no longer a problem. So usually the females will land on the carcass, and males wait around in order to find themselves a mate. And once the male transfers the sperm into a female, she then holds it in the structure called spermathechia. So the eggs are being actually fertilized while they're being laid, and they pass through the ovipositor. So most of the forensically important species that we find in Britain are oviparous, and they lay eggs, and the numbers largely depend on the species. With, for example, Lucyilia sericata can lay between 200 and 300 eggs in one batch. Occasionally, however, as the single egg gets fertilized inside the female, the fly may actually lay a single larva, because the eggs start developing inside the female. And this is quite an important thing for the forensic entomologists to know, because they usually collect the largest larvae in order to indicate the post-mortem interval, or what is commonly known as the time since death. So if in this case the largest larva is being collected, it may give the wrong indication of the time since death, because obviously it has been developing before it has been laid. So the flies usually choose sites that are moist because the eggs and the larvae are prone to desiccating. So the normal choices are all the body orifices, like mouths, nose, eyes, anus, and any cuts or other wounds. So we can see here the huge egg patches on the underside of the wild boar. So those pictures are taken in Poland. They've been part of my project that I did for my master's course on the composition of large mammals in the forest habitat. So the focus was actually on the impact of other animals other than the insects, but the insects were definitely the most commonly found ones on as feeding on the carcasses. So if we look closely, the adult blowflies are, in this case, are feeding on the secretions from the blistering skin on of the carcass of the wild boar, while the eggs are actually being laid in more shaded areas. And if we look on the other side, this white what looks like a substance is actually batches of eggs that have been laid on the wild boar's head. So those are pictures taken just a day after. Please disregard the date on the actual pictures. And we can already see, as mentioned before, the egg batches, but also the freshly hatched larvae. Different species take a different amount of time to develop, and this is highly dependent on the temperature. So some species are more develop better in the higher temperatures, and some of the species prefer lower temperatures. So let's say Lucyilia sericata, which is the green bottle, is the species adapted to high temperatures, and at 34 degrees Celsius, the full development can take just over 10 days. While, for example, Caliphora vesina, which is the blue bottle, would never develop in such high temperatures. Actually, the threshold for Caliphora, the developmental threshold is much lower, and it actually can develop in temperatures as low as about 1 degree. So as long as we are above freezing temperatures, they can actually develop. And as mentioned earlier, that the flies can actually indicate the position of wounds when you examine the carcass. The picture on the right, this is the site of a gunshot wound, because the wild boars have been killed by the gunshot. So this is just a day after the carcasses have been deposited in the forest. So let's talk a little bit more about the life cycle of the blowflies. As already said, once the eggs are being laid, it takes various amount of time, depending on the species and the temperature, for them to hatch and develop into first instar larvae, which we can see here. So while the larvae feed on the carcass, they stay their whole feeding time, they stay on the carcass, and they go through 3 developmental stages that are quite distinctly different from each other. And at each of those stages, they have mouth structures and some other structures that can actually allow for their identification. There are some very good keys that have been published by Krzysztof Szpila that I truly recommend if anyone is interested in having a go at identifying the blowflies. You will, however, need to slide mount them, so that can be quite a small obstacle. So the larvae feed on the carcass. They go through 3 different stages. Once they finish feeding, they then empty the gut, and at this stage, the larva is called a prepupa. And then they begin wandering. They move away from the carcass in order to pupate. So they usually dig themselves into soil, and their cuticle or the skin of the larva hardens into a shell of sorts that's called a puparium, and that shell protects the pupa while it rests and develop in the soil. So here is the already empty puparium, pupa case of Lucyilia sericata, the same species as the larvae we've been seeing in the previous slide. So once the fly is fully developed and it's ready to exit the shell, the structure on its head, called ptylinum, fills up with the liquid, and it explodes, sort of like the airbag in the car, and that opens the case so the fly can emerge. And at first, the adults are quite colorless and can be very difficult to identify, but within a few hours, they gain their normal coloration, and the wings that have been quite folded and crumpled, they start to fill with the liquid and extend. And after a few hours, the fly is ready to fly, and again start the whole cycle all over again. So females looking for protein food and males looking for females, and everything goes on. So the mentioned ptylinum, it retracts, and we can see the how it actually looks in the adult fly. There is a feature called the ptylinous suture that is kind of a remain of the original ptylinum. Talking about the forensics, there are various ways in which the caliphorids can be useful in forensic cases. The main ways in which they're utilized is in estimating the post-mortem interval, or the time since death. And there are 2 main methods that are being used for that. So one method is based on the development of the fly. Basically, a forensic investigator will collect larvae from the body, take them to the laboratory, and measure them, or some of the studies have been conducted based on the weight of the larvae, and based on that and the temperature, and looking at the previous studies done on those species, they can make the estimation of how long the larva has been feeding on the body. The temperature is usually taken at the site for a few days, and then compared with the temperature at the nearest weather station. And their linear regression is conducted in order to see the relationship between the temperature at the site and the weather station, and whether there is a good match, and it can be used for the estimation of the post-mortem interval. The other method, which is not as accurate as the development method we've been just talking about, is the succession studies. However, that requires quite a good research conducted in the same habitat and in the same climate zone on the species that frequent the carrion. So the very good study of the woodland has been conducted by Matuszewski in Poland. This is the study that I used in my master's project to compare to with what I could see happening on the carrion in the forest, because different stages of decomposition will attract different insects. So blowflies are usually the first ones to appear on the carcass, and they're first colonizers of the carcasses. And then there come succession of waves, including some carrion beetle species and other insects that actually feed on the larvae on the carcass. But as said, it's not as good as a method for estimating of time since that as the developmental studies. Of course, not all of the species have been thoroughly studied. However, quite few of the British ones have been, such as Lucyilia sericata and Caliphora vicina, Caliphora vomitoria. There is still no data for Lucyilia ampullacea, and Lucyilia caesar has only some of the unpublished data. Yeah. So looking at the distribution of the forensically important species, I would divide them into species that are widespread, such as the already mentioned Caliphora vicina vomitoria, Cynomyia mortuorum, which is somewhat more common in the north and not as common and quite scattered in the south of Britain. Lucyilia sericata, which is a widespread species, although till not so long ago, I've been told that it's not as common in Scotland. However, there is more records coming in nowadays, so that has been a bit rectified. Lucyilia caesar, which is probably one of the best known one species in Britain, however, quite a difficult one to identify, because it's very similar to Lucyilia illustris and Lucyilia ampullacea. So identification of those from the pictures can be very difficult and a real challenge. And in those cases, taking specimens is always advised. There is also Lucyilia silvarum that is also widespread but very scattered. Also, the preferred food source for this species is actually amphibians, similar to Lucyilia buffonivora. However, in this case, it causes the internal meiosis, so the larva actually feeds inside a living toad or frog. But it also will utilize carcasses of the vertebrates, and it also has been found in one case involving a human cadaver. Protoformia teranovia is also quite a widespread species, again a scattered distribution. I usually find it early in the spring because the species overwinter as adults, so they're usually quite early to come out. Then in terms of overall distributions, there are localized species, and we'll have a look at them more closely later on. And quite the weird ones, there are single records of 2 species. Formia regina, that is the species that occurs in every key to British Caliphorids. There have been multiple specimens in the collections. Sadly, all of them have been misidentified. So the status of this species leaves a lot to discussion. And Lucyilia cuprina, we're also going to have look at this species. There has been only a single record of it from Britain, and it's quite an interesting one. So please bear with me. I must say the recording of Caliphorids has been slightly neglected on my side. And this is mainly because I spend most of my time working on the identification key, but I do promise things are picking up from now, and hopefully soon we will have proper distribution maps and perhaps a atlas eventually. So Protoformia teranovia, here's the map that I've made not so long ago, based on the records that have been sent to me as the spreadsheets. As you can see, there isn't much at all. And it's extremely scattered, and I was very surprised how uncommon it appears to be. So there is mainly a lot of records in Scotland. And this is because there is a very good recording group working there, with which is organized by Murden MacDonald, and they kindly shared their records with me as well. But if you have a look at the records on iRecord, there is actually a little bit more nowadays. It's still not what I would expect. And to compare that with the NBN data, which comes from many different sources. So it's not only data from the recording schemes, but also from local record centers and some individual recorders. So it's not always properly verified. And yeah, we will see that later on as well. So talking about a bit more localized species, Lucyilia richardsi, mainly southern distribution. As we can see, it only has been recorded from Scotland on a single occasion, and this is the record coming from MacLeod and Donnelly study of 1954. So it has been a very big study across the whole of Britain, where they set traps baited with dead rabbits, and they had about 50 sites across the country, and they collected different species and created the distribution maps from that. And it's a very nice study because the specimens from it have been properly identified and properly vouchered, and the distribution that they showed, it actually shows that it's mostly the same nowadays. Speaking of Lucyilia richardsi, it is also of quite a limited value to forensic studies, because the species predominantly breeds in small mammals such as voles and mice. So it is not attractive to a larger carrion such as the human body would be. But because it is a carrion breeding species, and there is always a potential of it being found in forensic context, it is also included in here. The interesting thing about this species is also that it does occur in open habitats. It does not like the woodland. So, yeah. The other quite localized species, which is scarcely recorded, as you can see on the map from the iRecord, is the Caliphora uralensis, and this species is predominantly found on the shores. So MacDonald reports that most records are within 50 to 800 meters of the shore. And most recently, it has been recorded from the Bass Rock by Donald Smith, who actually was catching the flies with his hands as he was not equipped with a net. So thank you very much for such a devotion, and looking forward for more records of the sort. It has been also reported that Caliphora uralensis prefers to breed in liquid feces rather than the carcass. However, it is also reported to breed in carcasses of seabirds, and it's commonly found in relation to the seabird colonies. Another not very well recorded species, Caliphora luvoid. It is a bit more spread out than the previously discussed species. And predominantly, again, northern England, Wales, and Scotland. And it's believed to be preferred woodland habitat in the sites where it occurs. The interesting fact to point out here is that the coloration of the face of this species is variable. Many keys use it as the identifying character. However, the specimens with the orange coloration have been found in the collections as well. The two species that are very much like are the Caliphora stilviana and Caliphora subalpina. And it's quite interesting to see how they divide the Britain between each other. So their Caliphora stilviana is probably the rarest of our species. It's only found in the mountainous habitats over 500 meters above the sea level, and that only in Scotland and northern England. And where in those areas, Caliphora subalpina appears in the lowland areas. So, however, when we move to Wales, it takes over in the highland sites where Caliphora stilviana does not occur. It's quite interesting because Caliphora stilviana being probably the rarest species is actually quite well recorded when comparing to the others. So if we have a look at the map, we can see the yellow dots are the records of the Caliphora stelviana, and they are all in the mountainous habitat. And the purple dots, those are the records of Caliphora subalpina. And if you zoom in on Scotland, you can actually see they pretty much avoid each other in terms of the occurrence. So I don't think I've got a single record of both those species from the same site. So those kind of maps that you can see here, they have been actually created as part of my PhD project using ArcGIS, and hopefully in the future this kind of maps will be created for all of the Caliphorid species. You can have a look here. There are two records that do not fit with the with what we generally know about Caliphora subalpina, and there is one in Cornwall, which actually turned out to be a mistake in georeferencing. So the species was identified correctly, but during the transcription, the site name has been changed, and the site that has been georeferenced is not where it should be. And there is one more record, and this is actually a misidentified specimen, which I could track back to the actual entomological collection and verify that it has been misidentified. This is not a glitch in species occurrence. This also shows how important it is to actually keep vouchers of the entomological material, because while recording using the spreadsheets and even in some cases pictures, it's not always possible to verify the species identification, and some like very important problems can arise from that, especially if we're looking in the context of the forensics, which obviously can decide of somebody's fate. So there are two cases that I would like to have a look a bit more closely at, and this is Formia regina, which, as we said, occurs in every handbook and every identification key for British species. However, the specimens in the collections do not reflect that. So the Van Emden records the species occurrence as Oxfordshire. However, when I visited the collection, the specimen is no longer there. So the verification of the identification was not possible. On the other hand, there were quite few specimens from the Liverpool Museum. I'm sad to report that all of them have been misidentified, and in fact were Protoformia teranovia, I think most of them. There has been a single specimen reported from the Cardiff Museum, and that also turned out to be a different species, in this case actually a musket, so not a caliphorid at all. However, two specimens of the species have been found in the Edinburgh Museum, originally identified as Protocaliphora azurea. So completely different species, and those have been collected in Cambridge in 1915. So there is some record of this species actually being present in Britain in the past, but no recent records, no specimens have been collected, as far as I know, except a single study involving forensics. So a few years ago, there has been a study published by Green and colleagues, and that study was on the development of Formia regina, and the study was conducted in Britain. However, there was no information regarding the source of the specimens that have been studied. So I actually contacted the authors, and I found out that the specimens of Formia regina have been collected during spring season in Essex, which sounded quite suspicious to me, because the species that have not been seen in Britain since over 100 years suddenly appearing in Essex is suspicious at most. And how it turns out to be, I requested the specimens to be able to confirm the identifications. Sadly, they have not been retained. They have not been vouchered, so I cannot tell what species has actually been used in that study. From the information provided, I would guess, but this is only a lucky guess, that these species may actually have been Protoformia teranovia. But until the specimens can be located, there will be no confirmation. And quite interestingly, if we look at the map that I got from the NBN, the species is still being recorded. And similar as with the article published, there is usually no specimens. There are just human observations. There are no pictures. So the records stay in. They are unconfirmed. But as a matter of fact, I'm 99% sure those are just misidentifications.
There's one there that looks like it might be Rexon or something, with a pink dot above the word Stoke.
Yeah, there is one there, and there is one near Newcastle.
Yeah, I don't suppose you, because there's people on the call who may know about that. In fact, conceivably could have been the people who identified or misidentified that insect. So
if there are, please speak up.
No, you don't know.
Yes, I can. It's quite a justifiable mistake, because if one doesn't have vast experience in identifying the blowflies, and as we can see, it can be quite similar to some of the other species. So if you see something greenish blue that look like this, it can easily be recorded as Formia regina, and not many people actually go and read the scientific papers or read the books to learn about what the status of the species is in the country.
Yeah. Where will you have got that record from, though, Olga? Where will you pick that one up? How will you have got that record?
So those records on the map, the map actually comes from the NBN Atlas.
Okay. All right. So the source could be anywhere, really.
Yes,
okay.
It's not iRecords, because iRecord is verified by me. From what I remember, it's one of the local record centers.
Yeah.
They're not always very happy about me approaching them regarding identifications.
Okay.
Well, or Justin, they could have brought their specimen into the museum in Liverpool and compared it with our wrongly identified specimens. So
yeah.
We could be at fault there as well.
Yeah.
Not anymore, because all the identifications in the Liverpool are now corrected, especially in regards to forensic species. So
thank you.
No more justification there. So there is one more species that I would like to bring to your attention, which was an extreme surprise to me. there has been one specimen of Greenbottles sent to me among other specimens from Glasgow Museum. And looking at it, I just thought, no, this cannot be. This is impossible. The specimen was identified correctly by the student who actually collected it in Tolkien, which is close to Carlisle. The specimen is of Lucyilia cuprina, which is a warmth-loving species, which till not so long ago was not even found in Europe, only reported by Rogness, from around the same time as this specimen has been collected in Tolkien. We know that in Spain there is a population that keeps growing. There is not much known about cuprina in Britain. It's quite a surprise because the temperature is definitely way too low for it here. And I actually went to the site to check if maybe we can collect some and see if it's possible or not that the species has survived. So this specimen was collected in 1991. There used to be a group of students doing their, I believe, master's research or undergrad research at the farm. And that's how this specimen ended up in the Glasgow collection. I went with my family on the way from holiday. We stopped at Tolkien. We went looking for the farm. Turns out the farm sadly no longer exists. There is a housing estate in its place. However, the local pub owner did remember the students actually coming to visit the farm. He didn't really know anything about the flies, but still nice to confirm that this is actually the site. What has it been doing in Britain? It's hard to tell. I know there has been research conducted on this species in Weybridge, which is far south, and some specimens have been sent live to Durham. Although that research, from what I recall, was done in 1950s, so there is no way the species would survive for that long in the wild. There has been other research conducted in Durham. I'm not really sure if they kept the live specimens there of Lucyilia cuprina. It has been done by the probably known by all of you, Zakaria Ersinkoglu. So he's the author of the Blue Book, the Blue Key to Caliphora. Could it travel from Durham all the way to Tolkien? It's hard to tell, but how otherwise did the warmth-loving species recorded from hot climates end up in northern England? If you have any suggestions, I will be happy to hear. I will be happy to hear them. The other thing is that the species may have been actually overlooked in Britain because it's strikingly similar to our one of most common greenbottles, Lucyilia sericata. So you really need to look at quite a detail to be able to tell them apart. And if that was not enough, they also have been shown to interbreed in the lab conditions. So they may actually be all mixed up. And the hybrids that they produce are also fertile. So they're, apart from actual Lucyilia cuprina and actual Lucyilia sericata, there are also mixtures that can be encountered. Looking at all the maps that we have, it's quite easy to see that there is somewhat of a recording bias. So if we look at the clusters of the records, they usually are not so much where the species are common, but where the recorders are common, or where the recorders go on holiday. And we still have quite big patches of Britain that are not well recorded. Also, we may expect that the species that are difficult to identify or that they will be somewhat less recorded. But in fact, what I've been finding is that oftentimes, as we could see with the Caliphora vienna, those are actually species that people do focus on, which means we have more records than if we look at more common species. Also, most people don't bother recording Caliphora vicina because it's the species that they see almost every day and almost everywhere. But I think the message that I would like to send is that it is very important that the common species are being recorded as well as the uncommon ones. Because when we later look at the records, or somebody who is not as accustomed with the British species may actually get the impression that those are the rare species. The other thing is that what I see looking on iRecord is that the records quality vary greatly. So there is still a tendency of submitting records that are not supported by any visual aids, so to speak. And unless I know that the recorder keeps the specimens or donates them to your collection, it's very difficult to verify them. And yes, how can we trust a record when it actually cannot be verified, even if it's a common species? So what can you do to help us? Help record the distribution of blowflies, not just the forensically important ones, but all the others as well. Submit your records, submit your sightings. Mainly, well, the easiest way is to do it through the iRecord. I would kindly ask you to attach the pictures if you have any, because that makes things much easier and much quicker for me to put the records through, and that makes populating those maps much easier. I'm quite happy for the recorders to post the specimens to me for the verification or identification or whatever helps. It may take some time for me to come back to you because there is always a lot to do and not enough hours in a day. And just be aware that some of the species that are common may be actually very difficult to identify from photographs. So I already mentioned Lucyilia caesar species groups, which is Lucyilia caesar illustris and Ampullacia. Those are very difficult, and most views that people photograph, they will not allow for identification to the species level. And then recording the species only at the genus level only gives you so much information. And then how useful is it for the researchers or for community? hard to tell. I would argue that it's probably not that useful and not well spent time, but I'll accept all of the records in hope that one day somebody may put them to a good use. Keeping vouchers is probably the best idea because they are constant changes in the taxonomy. So some species may be split, some species may be redefined. If you don't have a specimen, then that record may get outdated and it can be impossible to actually tell. So the Melinda, for example, they used to be recorded as one species. Now they are two species. In the study that I mentioned, conducted in 1954, they actually mentioned that Caliphoras have been split to a great extent. So most of the records previous to that are if they were not supported by the voucher material, are actually no longer useful. Also, I kindly invite you to visit our Facebook page and join it if you're interested in all things blowflies. Well, we do share quite a range of pictures and tips on identifications, biology, anything that you might be interested. Surprisingly, we are being joined by quite a lot of people from outside of Britain. So occasionally we do have requests for identifying species from, let's say, Korea or Africa, which might be quite interesting for you to see as well. And also there is a website that you can visit, linked to Dipjis Forum. And on that website, there are currently two posters that are a free download. One is the general poster for recording scheme that also gives you a few species that are readily identifiable from the pictures. So the species that I would very much welcome to be recorded in that way. And there is also a key to subfamilies, genera, and species of forensic importance. So exactly the flies that we've been talking about today. And thank you so much for listening. I would like to also say thank you to quite a few people who provided the photographs and the records so far, who shared the specimens, and the local record centers. Some of them do share their records with me, and especially the Highland Biological Records recording group, because they send me all of their records that have been verified and collected by Murdo. My supervisors and the Natural History Museum in London, where I'm usually based, and the Angela Marmont Center, which kindly provided their photographic equipment to take all the lovely pictures of the specimens, and also all the people at the museums that I've visited so far and taken the records from. Those records will soon be put into a good use. So hopefully we can maybe do a catch-up of this talk in about a year when I get all the beautiful maps ready for you to see. And yes, to my dear husband who patiently supports me throughout all this research. Thank you so much.